Hello, everyone. Welcome to the Duncan Comprehensive Cancer Center webinar on young adults and cancer. My name is Victor. I'm a second-year medical student at Baylor College of Medicine. Cancer is a topic that is a bit of a weak spot, but this is the reason why I push forward. What happened to my brother was not fair, and instead of hanging up my coat, I'm going to do something about it. Some things are out of your control, but making a difference is not. So it is my sincere pleasure to moderate with y'all. I really thank you for this opportunity that I do not take for granted. It is my pleasure to introduce to you Dr. Kent Osborne, founding director of the Duncan Cancer Center and breast oncologist.
Thank you, Victor. Welcome everybody to our webinar on cancer and young adults. This slide shows the three speakers that we're going to have tonight. I used to be the director of the Duncan Cancer Center. Now I'm the founding director of it. I stepped down from being director last month. Joe Petrosino is one of our speakers. He's the chair of molecular biology and microbiology and the head of the Center for Metagenomics and Microbiome Research. And then also we have Ji Yoon, assistant professor in the Department of Molecular and Human Genetics, who's going to be talking about a very interesting subject about sugar and cancer. Joe's going to be talking about microbiome and cancer, and I'm going to give an overview of cancer in young adults. Also, we have at the end, we have three other panel members. Dr. Gustavo Rivero, who's from our hematology group. He's an expert in hematologic malignancies, which are relatively common in young adults. Kathy Sullivan, who's head of our genetics program, and Ben Musher, who's an expert in pancreas and colon cancer. And you'll hear a lot about colon cancer tonight. So I wanted to start by talking about two cases that you've heard about, one of them at least you've heard about recently. And that is the actor Chadwick Boseman. He was a 43-year-old African-American male, and he recently died of colon cancer. No one even knew he had it. He was even acting shortly before his death, but he died of colon cancer. The second case is a 4-year-old Jewish woman who happens to be the mother of two of my three grandchildren. And she was diagnosed recently with colon cancer and just finished her chemotherapy. So the question is why? Why did these two people get colon cancer at a relatively young age? We didn't even start screening until age 50. Now it's reduced to age 45. But why did these people get colon cancer at such a young age? So let me talk a little bit about why some of these people get cancer at a young age. This is a picture of a normal cell. It has DNA in the nucleus. That's what these strands are here. And the DNA encodes proteins that provide a normal function to that particular cell. Our cells are bombarded with carcinogens all the time. And they can cause, if they escape the repair mechanisms that I'll talk about in a minute, they can cause mutations in the DNA such that a cancer cell is formed, and then we have a diagnosis of cancer. Luckily, normal cells and cancer cells are like a little village. It takes a village, you've heard that saying before. Villages have a city hall, they have stores, they have gas stations, they have all kinds of things to make the village. Well, cells have the same kind of things in a micro scale. And they have these little factories, I call them, that repair DNA from the DNA damage that's occurring in our cells all the time. If we didn't have these little factories, you probably wouldn't even be born because you'd have mutations during fetal development. So these DNA repair factories repair the DNA and prevent cancer. But what happens if you get a mutation in one of the factories so that the factories don't work right? And now you get a bunch of mutations in several genes, and now you have multiple mutations. And because of that, you have cancer at an earlier age in life. So maybe a breast cancer patient who has a hereditary breast cancer gene in BRCA1 or 2, those BRCA1 or 2 are factories that repair DNA. If they can't repair the DNA, then they get breast cancer when they're 30 or 40 rather than when they're 60 or 70. So the causes of cancer and causes of mutations are as follows. Cancer is really a sort of a combination of environmental exposures, perhaps, and damage to the DNA or mutations. This can be caused by chemical exposures, products of metabolism in our cells, exposure to radiation, chronic inflammation, or inherited mutations that cause these early onset cancers in families, like I just described. My daughter-in-law had such a mutation. We didn't realize that until after she developed the colon cancer, and we screened her for mutations, but she had a mutation that's relatively common in Jewish people, and it gives a 2 to 3-fold increased risk of colon cancer at some time during their life. Here's a list of some of the cancers that are more common in young adults. Brain tumors are more common in older people, but in terms of cancers in adolescents and young adults, brain tumors are one of the more common type of cancer. Lymphoma and Hodgkin's disease. The classic presentation of someone with Hodgkin's disease is a young adolescent woman who comes in with fever and night sweats and has a large mass in the chest. That's a classic diagnosis of Hodgkin's disease. Breast or ovarian cancer can also occur in young people. The youngest I've ever seen was age 20. And those people generally harvest a mutation in one of these repair genes like BRCA1 or 2. There's now a whole host of others, other genes that when mutated can cause breast cancer and other cancers. Colon cancer, as I mentioned, is becoming more and more common in young adults. We don't quite understand why. Is it genetics? Is it environment? Is it microbiome? Is it sugary drinks and sugar? You'll hear more about that later. Cervical cancer is relatively common in young women, and almost all cervical cancers now are due to the virus, human papillomavirus. That's why all of our kids should be vaccinated against that virus to prevent this cancer later in life. Testicular cancer occurs in young men. Thyroid cancer tends to occur in younger individuals, sarcomas, and melanoma because of sun exposure. And also, there are mutations that cause an increased chance of getting melanoma or a sarcoma sometime in your life. So how can we prevent these cancers in young adults? Young adults are not walking around thinking they're going to get cancer, but what do they need to do to help prevent it? Well, obviously a healthy diet. Obesity is a major cause of cancer and other problems now that we're dealing with. It's an epidemic in this country, and having a healthy diet will help to avert that problem. Minimize alcohol. Alcohol increases the risk of breast cancer. Many people don't realize that, but it also increases the risk of esophageal cancer, head and neck cancer, and other cancers as well. Maintaining an ideal body weight is very important. That's a result of a healthy diet. And even vitamin D may be playing a role. Vitamin D we think of as being important for bones and strong bones, but there's also probably some anti-cancer effects of vitamin D. And just as an example, 50% of the general population in this country are vitamin D deficient, while 70% of breast cancer patients are vitamin D deficient, suggesting at least that maintaining a vitamin D level in the normal range would be very important. Exercise is also important. It's been recommended that 30 minutes a day or 3 to 5 hours per week of walking can reduce the risk of breast cancer. Avoiding tobacco should be obvious in terms of its risk of lung and head and neck cancer and other cancers as well. There's 7 different types of cancer that are increased in smokers. Avoiding sun exposure for melanoma and other skin cancers. HPV vaccination is very important for young people. And then genetic testing in those who have a family history of cancer, like breast cancer, colon cancer, ovarian cancer, melanoma, thyroid, or prostate cancer, particularly if any of these cancers occurred in a relative who is relatively young, less than 50 years of age. And finally, in some cases, we do prophylactic surgery in the case of certain mutations that cause a very high risk of cancer. BRCA1 or 2 cause a 70% risk of getting breast cancer. So some women choose to have prophylactic mastectomy to reduce that risk. There are some genes that cause multiple polyps in the colon with a very, very high risk of colon cancer. And some of those patients choose to have a colectomy to prevent that risk. What about screening? Obviously, when you're 30 years of age or 25 years of age, you don't think much about getting screening or even having an annual physical exam. But that's still an important thing, even for younger people. A testicular self-exam for males, even in adolescents and very young adults, high school age kids. Skin exam for changing our irregular dark moles. And you can learn what a bad mole looks like just by Googling it. Colonoscopy at age 45 or even earlier with a family history. As I mentioned, it used to be recommended to start at age 50, but because colon cancer is now becoming more and more common in younger people, they've lowered the age to start screening. A mammogram with a family history of breast cancer. Even if you're gene negative, if you've been tested and you're negative, and that mammogram should start about 5 to 10 years younger than the youngest affected family member. So if your mother had breast cancer at age 50, even if you're gene negative, you should start screening at age 40 and so on. And screening with a breast MRI is more sensitive than a mammogram if your risk is calculated to be high enough. And you can use a risk disc or other models of risk. There are computer models that are very simple to use that calculate your risk of breast cancer to age 85 or 90. So to summarize my introduction, cancer is caused by environmental and genetic factors leading to mutations in the DNA. It's uncommon in young adults compared to older people because it takes time for mutations to occur, except with hereditary susceptibility gene mutations like BRCA1 or 2. Also, as we get older, those DNA repair factories that I mentioned function less well, and that allows some of the mutations that normally would be repaired to escape and cause cancer. Some cancer types are occurring earlier in life than in the past, like colon cancer. We need to educate our children and young adults about prevention and screening. And obviously, prevention and early diagnosis are key. So let me now turn to our first speaker, Joe Petrosino. And Joe's going to talk about the microbiome in cancer. And what is the microbiome? Joe, go ahead and tell us all about the microbiome.
Thank you, Kent. Can you see my screen?
Yes.
Okay. So it's a pleasure to be able to speak to this group tonight, and hopefully I can give you a few take-home messages that will hopefully stick over the long haul and may impact how, well, hopefully you won't have a cancer episode in your life, but will help with friends or family should it occur in terms of the potential future of where certain diagnosis and therapeutics may arise with relationship to the microbiome as a cancer intervention. So let's see if I can advance this. There we go. So just a couple talking points, just to kind of guide us in the direction we're going. So I'm going to start by talking a little bit about the microbiome itself and what does it do. The microbiome has been in headlines for a variety of reasons over the last 10 years or so, and so it's becoming more and more familiar in the general public. But just to make sure we're all on the same page, I'll talk a little bit about that, a little bit about the research that we're doing at Baylor College of Medicine, and then with respect to cancer, a few vignettes regarding a few different types of cancer and cancer treatment, and then to go over the diagnostic and therapeutic implications going forward. So to start off with the fact that many of us are aware of, I hope, is that we are colonized by trillions of bacteria all over our bodies, both inside and out. And these microbes are referred to commonly as the human microbiome. The microbiome actually outnumbers the number of human cells in our body. We don't see them because they're much smaller than our human cells. But if you wiped them all off somehow, internally and externally, they would weigh about 3 to 5 pounds. Not a good way to lose weight, but nevertheless, they do contribute to our body mass. There are about 100 times more genes encoded by these bacteria on us and in us than the genes that are in our own human genome. So if you think about that for a moment, you heard Dr. Osborne a moment ago refer to the cell as a village with all sorts of functions encoded by the genome, telling the body to make certain proteins in every single cell. Well, there are 100 times more genes available to the bacteria in your body just because of the diversity of organisms that are there. So they encode a lot of functions that we as humans don't have. And as a result, actually functions that we need to live. And so the human microbiome can be thought to live as a symbiont on the human body, providing benefits for us while we also provide benefits for those organisms, a nice niche for them to live in. So I think of the microbiome. So another way to think of the microbiome is sort of our own personal accessory package. We have, again, the human genome encoding many different functions that keep us alive and make us who we are. But the bacteria in our bodies, the good bacteria, provide a lot of functions that we either need help with or wouldn't be able to do without them. And so some of them are listed here. Breaking down food properly, especially vegetables, requires the work of bacteria. Break down those hardy cell walls of plants. They provide, they enable our bodies to regulate the level at which we store fats. They promote blood vessel growth. Importantly, and you'll hear about this more as we get into cancer, they're involved with the development and the maintenance of our immune system, basically how well our immune system functions, and keeps it in at a balance level where it's not overreactive to the environment and other bacteria and stimuli that it may see, but not underwhelming either. So it is able to defend our bodies against pathogens. So our microbiome is key as a key component of that development. It also produces a lot of vitamins and amino acids that our bodies cannot produce by itself. And so, again, this goes on, but you can get the picture that the microbiome is an important component of our well-being. And I sort of think of it too as like taking something that's already cool, like a Ferrari, and then putting jet engines on it. So it's that type of benefit that the microbiome provides to us. So in realization of this, about 15 years ago, the National Institutes of Health launched an initiative to learn more about the microbiome at a national research sort of level. In that they started a program called the Human Microbiome Project to be able to create and develop the technologies that researchers will need and clinicians will need to be able to study and assess the microbiome as it relates to health and disease. And so in response to that, in 2010, we started the Center for Metagenomics and Microbiome Research here at Baylor to do translational microbiome research. That is, learning more about the microbiome, exactly how it is impacting health and disease, and to be able to develop new therapeutics and diagnostics that we can then bring to patients and help treat a variety of diseases. And so the model of our center is indicated here. And I won't go into each component, but we have also started launching companies in this space too, to be able to help deliver these types of research tools to pharmaceutical companies to further quicken the development of new tools and diagnostics. And I'll also launched a company called Anasome, which is actually using these same types of tools to look at the microbiome and how it could benefit companion pets such as dogs and cats. So when I mentioned the fact that the microbiome can have an impact on a variety of diseases, I'm not kidding. So over the last 10 years, we've engaged over 200 collaborators from around the world and in over 400 different projects involving any disease that you could probably imagine. Some of them I've never heard of, including those involving cancer. So specifically around colon cancer, immunotherapy, and pancreatic cancer. And the starting point for a lot of these studies, and some of the studies I'm going to talk about in a moment, some of which we were involved with and some of which we weren't, involve starting with an easy comparison, taking patients that are sick and taking the microbes from a specific body site, often the gut, and then comparing them to those who are healthy and comparing to look at which bacteria are present, which are present in healthy individuals, which are present only in sick individuals, and what functions that are encoded in those sick or healthy bacteria that may be contributing to the disease. Once we identify those organisms, we can then study them to identify which specific strains of bacteria may be most important in those health or disease states, and then identify whether it's a community of organisms, a subset of a consortia of organisms, a single strain, a metabolite, what exactly is contributing to the disease so that we can create new diagnostics and therapeutics that can benefit human health. So as we take it to cancer, I think the important message that I would like people to take home is the fact that microbial signals, the ones we just talked about benefiting health and disease, can also impact cancer onset, progress, and resolution. And this is sort of a complex figure, but in the internal component, we have bacteria and bacterial products impacting a variety of processes in this tan sort of outer ring that are known to be associated with cancer development. And so we don't have time to go through each and every one of them here right now, but safe to say that microbes have a strong impact on, a strong potential impact on a variety of processes involved with cancer. So that's take-home message number one. And so because of these impacts on these various stages of cancer, we then have the ability to use these tools, use the bacterial signals or the bacteria themselves as diagnostic and therapeutic targets. And so you can imagine if there's a way to identify bacteria that are associated with a particular cancer, or even people who are susceptible to certain types of cancer, then that's another diagnostic tool that we could use to detect cancer earlier and thereby lead to prevention, as Dr. Osborne mentioned in the previous talk. So to kind of give an example of this, one observation that was made early on and has been delved into even further is the fact that microbes associated with colon cancer change with cancer progression. What I mean by that is that if we take biopsies from a patient with colon cancer at various stages of cancer development, and even those with benign polyps, and compare them to the bacteria of healthy individuals, and compared to other stages of colon cancer, we can identify unique microbes that are associated with each stage. And so as these organisms become more abundant in the gut, and these would be read out in a stool sample, you can potentially identify a patient who may have an issue before a biopsy or before a colonoscopy is even performed. And so these types of signals are the types of signals, the names of certain bacteria, and none of them are able to be pronounced easily, and microbiologists design them that way. That's how we keep our jobs. But by having organisms that are specifically associated with certain stages of cancer onset, we can potentially use these as diagnostic targets. And potentially, because they are associated with these different stages of colon cancer, examine if there is a causal relationship. That is, if we remove those organisms or remove the organisms that are associated with the next stage, can we arrest cancer development and prevent it from progressing further? So a vignette that I wanted to start with is a Microbiome Association study with pancreatic cancer. And this was done with our collaborator, Florencia McAllister at MD Anderson. And the question that we wanted to examine with her was whether or not the microbiome of the pancreas, a pancreatic tumor, has any kind of association with outcome from treatment for pancreatic PDAC, pancreatic ductal adenocarcinoma. And so the prognosis for pancreatic cancer, as some of you may be aware, is not very good at all. And so looking for new therapeutics, new diagnostics that could potentially identify pancreatic cancer earlier in onset could potentially have an impact on prognosis, on outcome. And so what we looked at in this case was that we looked at the tumor microbiome in pancreatic cancer. So the microbes actually living in the tumors themselves, comparing them to people that had a short-term survivor outcome and a long-term survival outcome, looking at the same individuals at the gut microbiota, so the bacteria found in stool samples collected from the same individuals, and then looking at the immune profiles of these individuals. So looking at the immune response, because we're now looking at a lot of treatments that utilize the immune response, and also knowing that the immune response has a specific direct impact on cancer progression in many cancer states, not just pancreatic cancer. And one of the things that we found right away, and so I am going to show a little bit of data, but I'm going to walk you through it so it's not too daunting, is that we did find a signal associated that differentiated long-term versus short-term survivors. That is the number of species of bacteria found in the tumors of patients that had a long-term better outcome, shown in blue here. They had a more diverse microbiome, both in the tumor itself and their gut microbiota. And so that's all that's shown here on the left side. So comparing the long-term survivor microbes, number of microbes shown on the y-axis, to those that were found in the short-term survivors. So having a diverse microbiome, which is a sort of a theme you'll hear about with a lot of diseases or maintenance of health, is that a diverse microbiome is good for you. It's like maintaining a garden. The more different species of plants you have, the prettier your garden, the more robust it is. When we looked at the same samples a little bit more deeply, we identified very specific bacteria, again, unpronounceable, that these specific bacteria also were associated with long-term survival. And which was very interesting because, to this point in time, very few people, if any, would postulate that bacteria living in the tumor, A, were there, B, really meant anything in terms of overall health and disease. But the fact that we could identify them not only in patients at MD Anderson, but also in patients at Johns Hopkins University. So this signal wasn't just some sort of artifact we saw here in Houston. It was something that was reproducible in another location. And because of that, we went on to do further tests and show that if you take the tumors from patients and recapitulate the same experiments in a preclinical animal model, you can actually, if you take the short-term survival microbiome and put them with the tumor in, say, a mouse, that they would also have a poor outcome. However, if you took these organisms and put the microbiome containing these organisms in a mouse with a person that had a long-term survival outcome, that mouse would also behave similarly. And so with this information, we now have some targets that we can go after, both in terms of diagnostic. So people be undergoing treatment for pancreatic cancer, if they have these specific bacteria, perhaps they will have a greater chance for longevity past 5 years with these organisms. And over the longer haul, identify exactly how these organisms are impacting defense against the cancer progressing further. I can say right now, I also mentioned the fact that we're looking at the immune response at the same time. What is happening in patients that have these organisms is that the immune response is more robust at the tumor itself. The immune cells are infiltrating the tumor and they're helping clear out and destroy some of the cancer cells. And so that's why those patients with these organisms tend to have a better outcome, we think. And so more is to be done on that front. So this paper was just published last year. And I'm going to switch now to a paper that's published just the year before, looking at a completely different story, but not unrelated in this case, because we're still talking about immune system relationships in the microbiome. But many of you heard about immunotherapy. And so with immunotherapy, this is training the body to attack the tumor itself. So normally your body does not want to attack itself, otherwise you have autoimmune diseases. But if we can somehow allow the immune system to not recognize the tumor itself, then the immune system can attack it like it would attack a pathogen. And so there have been a variety of new and wonderful drugs called checkpoint inhibitors that have allowed this type of therapy to be done for certain types of cancers, melanoma and non-small cell lung carcinoma being just two of them. And so the question that we asked with Jennifer Wargo at MD Anderson was whether or not there's a microbiome signature that was associated with success or failure in these treatments. So again, immunotherapy, you want the immune system to attack the cancer. And so again, because the microbiome and the immune system has such an important crosstalk with one another, the hypothesis was that perhaps the microbiome is associated with success or failure of this treatment. So normally immunotherapy up until recently has had about a 30% success rates in individuals. And we compared the gut microbiota in responders and non-responders, just like we saw in the pancreatic comparisons. We saw bacteria that were associated in those people who responded to immunotherapy and those that failed immunotherapy. So responders and non-responders. And so again, you start off with sort of this observation, and then you need to understand, well, is this causal, or is this just an output? Is this a canary in the mine, which could be used as a diagnostic, but is not as interesting in terms of a potential therapeutic target later on. So what we did with Dr. Wargo is that we took tumors from responders and non-responders, and we put them into mice. And then we took the microbiome the gut bacteria from those same patients and put them in the same animals and treated them with the immunotherapeutic treatment and found that they had the same outcomes as it did in the individuals. The responders responded and the non-responders didn't respond. But if we took a non-responding tumor and put in a responding microbiome and then give them the PD-L1 or the checkpoint inhibitor, the non-responding tumor shrank, went away, showing that the responding microbiome had an impact that catalyzed or that helped work with the checkpoint inhibitor immunotherapy to make the tumor go away. And again, we see that the reason for this is because it allows the immune system to act more robustly against the tumor, as opposed to those who had a non-responding microbiome. And so this is now being worked on, developed in many different ways across the world in terms of developing new therapeutics to administer with immunotherapy to make the immunotherapeutic drugs work more effectively. So with these, just a few of these stories, there's many others that are like this with a variety of cancers. The question that you get often is well, how do I help maintain a healthy gut microbiome? And of course, the answer, as you heard from Dr. Osborne's point of view or presentation earlier, not only for our own health, but for our microbiome's health, it's important to eat a healthy diet. It's the number one way to keep the good bacteria. It's like fertilizing a garden. And so leafy greens, bacteria love fiber. And so that's another good reason to get your fiber. But at the same time, we and many other groups, again, from around the world and biotechnology and pharmaceutical companies are developing new therapeutics and diagnostics in ways based on the microbiome to, again, help with cancer and a variety of other diseases. And so with that, thank you. Some contact information, but feel free to reach out to me anytime, and we're dealing with questions later.
Thank you, Joe. Our next speaker is Jihye Yoon. She's in the genetics department, and she presented a really interesting paper a year or so ago in the journal Science, one of the top journals, about why are sugary drinks particularly bad for cancer development. And she's, I think, going to talk a little bit about that and sugar in general. Big issue right now for cancer. Jihye.
Yes. Okay. Can you see? Okay. Thank you, Kent, for invitation, and good evening, everyone. My name is Jihye Yoon, and our lab studies how diet affects colon cancer development using mouse models, and we study both bad diets and good diets. And today I'll talk about a bad diet, sugar, especially sugar drinks, and how sugary drinks can increase the risk of developing and dying of colon cancer. So sugar consumption, especially in the form of high fructose corn syrup, has dramatically increased since the 1980s. And during the same period, rates of obesity and metabolic syndromes such as type 2 diabetes, along with colorectal cancer among young and middle-aged adults, have also increased. And these links are not just association, and growing a number of studies now show that increased sugar consumption can cause many metabolic diseases, including cancer. So what is the high fructose corn syrup? High fructose corn syrup is a type of refined or added sugar that you can find easily in most of the processed food these days, and also is a main ingredient of sugar drinks. And it is a mixture of simple two sugars, glucose and fructose, about 1-to-1 ratio. And making high fructose from cornstarch was actually invented around the 1960s by a scientist. And it is much cheaper and easier to produce than extracting sucrose from sugarcane. So that's the reason food companies replaced the sucrose with high fructose corn syrup. And sucrose, which you can find in all green plants, is also made up of two sugars, glucose and fructose. But the difference is that these two sugars are chemically bound together, as you can see here. But this chemical bond can be easily broken by an enzyme called sucrase, that is secreted in our body, the small intestine. So basically, sucrose is exactly same as a high fructose corn syrup once they are in our body. So these days I noticed that food companies, including Coca-Cola company, saying that, oh, our new product is healthier than previous one because we use the sucrose from sugarcane, not the high fructose corn syrup. But they are actually fooling you. They are basically saying the lie, because basically high fructose corn syrup and sucrose are exactly same, and they are both equally toxic and bad for our health. So why did the world increase the sugar consumption dramatically in the past 2 decades? I think there may be multiple reasons, but it came down to the one reason, and that is the greedy in sugar industry. And the greedy is exactly same as the tobacco industries, and they don't care about our health. They care only about their profits. So for example, 2016, the New York Times published an article showing that the sugar industry paid the scientists at Harvard in 1960s to play down the link between sugar and heart disease, and promoting saturated fat as the culprit instead. And the public believed that, and they did a campaign to reduce the fat in our foods. But when you reduce the food in our foods, the taste is like eating cardboard. So food companies had to put tons of sugars in our foods to make it taste better. And around that time, as I mentioned, the high fructose corn syrup was invented, and it is much cheaper to produce added sugar than before. So since then, sugar started invading to all of our foods, most of our foods. So also, sugar companies aggressively advertise their product, especially sugar drinks, targeting all ages. So as you can see in this 80s advertisement, they basically want to sell this toxic substance to even toddler, and they know that sugar drinks are bad, but even so, they actually sell, which is unethical. And worst thing is that sugar is highly addictive, like a cocaine. And I can talk a lot of things about sugar in our health, but I want to focus on today that bad effect of sugar in colon cancer. So generally right now, well accepted that sugar can contribute to the colon cancer through the obesity. So if you eat excessive amount of sugar, it will cause obesity, and obesity is a well-known risk factor of colorectal cancer. And this is a correct view, but I want to point at the one kind of misconception that some people have, which is some people think that nutrients from fat or proteins can also cause equally obesity, as long as they consume same calorie as we consume sugar, which is not true. And our study actually in mice showing that sugar is much powerful to cause obesity and other metabolic syndromes, which I will talk shortly. And another misconception is that many people think that sugar cannot directly cause colon cancer independent of obesity. But our mouse study showed that even modest amount of sugar, drinking sugary drinks, can cause the colon cancer independent of obesity. So first, I will talk about the first myth is that all calories are created equal and sugar is just empty calories. And that's the kind of a mantra that Coca-Cola company and other sugar company want to promote as a truth. So basically what they're saying is that we can drink soda every day as long as we eat less of other foods and exercise more, because at the end, the total calorie, only total calorie matters. But that's not the truth, which I will show in our mouse study. So in our mouse study, In control group, we gave a mice water, tap water, and chow, which have starch as a carbohydrate, and proteins and fats and other essential nutrients, micronutrients. And in sugar group, we gave us exactly same chow to mice, but we replaced the tap water with 25% high fructose corn syrup in water. So, and we gave this complete freedom to mice to eat whenever they want, how much they want. And we measured the consumption of water and chow in these mice for 8 weeks. And we calculate the total calorie intake in these 2 groups. And we obviously, these sugar group mice drink a lot of water because they love sugar, just like we do. But actually, they are smart. They don't actually consume much chow, so which was actually a surprise. So when we actually calculate the total intake, it was actually almost the same between 2 groups. So if the Coca-Cola company is correct, I mean, they should have basically a similar weight. However, in this sugar group have dramatically increased the weight, as you can see here, and this sugar group obviously is bigger. So this sugar group not only have the obese phenotype, but also they have type 2 prediabetic phenotypes, and also they acquire the fatty liver disease. And this is not just the mice thing. I really recommend to watch this sugar film, which is free, I think, on YouTube and Amazon. So in this film, the director experiment on himself. So he maintained a similar total calorie, but he increased the sugar consumption, including sugar drinks. And even only 18 days, he gained almost 20 pounds, and he increased the waist. As you can see, the belly is bigger. And he developed the fatty liver and pre-type 2 diabetes and increased the heart disease risk. Of course, he stopped and go back to healthy diet. So this phenotype, this disease is reversed. But if you continuously do eat high sugars, I'm sure that it will be point that it will be not be reversible. So I will talk about now how sugar can directly cause colon cancer, independent of obesity, at least in mice. And in this experiment, we use the APC mutant mice. So APC is the kind of a gatekeeper in colon cancer. So more than 80% of colorectal cancer patients lose these APC genes by mutations. And when you don't have this gene, you will develop very benign precancerous tumors. So it's not cancer yet, but there is a potential to develop cancer. So we use this precancerous mouse model and give the same chow and water. And then in this sugar group, we gave this mice, sugar group mice, very tiny amount of high fructose corn syrup via oral baths daily for 8 weeks, and which mimic the less than 1 can of Coca-Cola in humans. And the reason we gave the very tiny amount is that we don't want to make this mice fat or increase the weight gain, because we want to eliminate the fact of weight gain here. So you see the direct effect of sugar. So when we give this tiny amount of high fructose corn syrup, mimicking drinking Coca-Cola one day, actually there is no weight difference between control and sugar groups. However, when we analyzed this mice tumor burden, we saw the dramatic increase of tumor number and tumor size and tumor grade in this sugar treated group. And we tested several different mouse models, and it all consistently show that this sugar directly actually increased the tumorigenesis and development. So what's the mechanism? How sugary drinks affect the colorectal cancer development? I will not go into the detail, but I will tell you the broad 2 possibility based on our studies. And one is through the direct uptake of glucose and fructose by tumors, APC mutant tumors. So when you give the sugar drinks in liquid, basically sugar in liquid compared to other cookies or cake in a solid sugar, this liquid form of sugar actually saturates much fast the transporter in the small intestine, and it goes spill over to the colon, resulting in high concentration of fructose in the lumen, colon lumen, and also increase the blood level in glucose concentration in blood. And these tumors actually have highly expressed the transporter to sucking up the all glucose and fructose compared to other normal cells. So that these tumors use these sugars as a fuel to continue to grow. That's the one way sugar drinks can affect the colon cancer. And the other possibility, as Joe mentioned, that there are a strong link between diet and gut microbiome and colon cancer. So we saw that when we give the high fructose corn syrup to mice, we saw the many bacteria chains that is actually correlated with the human colon cancer patient profile, which is telling that this altered microbiome can affect the colon cancer development as well. So what's the relevance of our study to humans? As Kent mentioned, that right now there are growing concern that more and more young people are diagnosed and dying of colon cancer, which nobody knows why. And based on our study, mouse study, and also human epidemiology study, although it's not quite as strong yet, but I think that one part of the reason we can explain is that of this young onset colorectal cancer is the high consumption of sugary diets, especially sugary drinks. And when you have these small polyps in the colon, usually it takes 20 to 30 years. But if you continuously consume the sugary drinks, this will shorten the time to become aggressive cancer. And not only the early stage of colon cancer, right now we are also looking into the possibility that sugar drinks can affect the later stage of colon cancer. And we have a preliminary data that high fructose corn syrup, sucrose can also increase the migration and invasion of cancer cells. If that is actually happen in human, we really have to be careful to give the energy drinks like Ensure or fruit juice to cancer patients, which is actually occurring in some of the places because to help the patient, but actually may be harmful, more harmful to cancer patients. So the take-home message of my talk today is that sugar drinks are new cigarettes. They both are highly addictive. They both are toxic to our bodies. They both affect our cancers. But the big difference is that tobacco, smoking is highly regulated by government. But sugar drinks and sugar are not restricted. Everybody can access, right? So I really hope that you can avoid the sugar drinks and importantly, try to not to give to children. I think that is actually important. You don't want to give the toxic substance to the children. So thank you for listening. And if you have any question, feel free to contact me. And I hope that you will learn something from our talks today.
Thank you so much, Dr. Yoon.
Mm-hmm.
We will now take questions from you. To help us give you most comprehensive answers, joining us is Kathy Sullivan, a genetic counselor with the Duncan Cancer Center, and Dr. Benjamin Musher, GI and pancreatic oncologist, as well as Gustavo Rivero, a hematologic oncologist on the Cancer Center.
Maybe I could start by asking a question. Can you hear me? Joe,
We can hear you.
okay. Joe, I can understand how the microbiome might impact cancers in the colon or near the colon, like the pancreas. But how in the world does it affect things like autism? I saw that on your list, or things that are far away from the gut. How does it do that?
No, that's a great question. And so systemic impact of the microbiome is really where the cutting edge, the tip of the spear is right now in terms of microbiome research. And so we know right now, for example, that much of the serotonin, for example, that's produced in the body, over 70% comes from the gut and travels through the bloodstream and crosses the blood-brain, is able to both signal cells in the gut, but also travel in the blood and cross the blood-brain barrier. And we also know that short-chain fatty acids such as butyrate also have impact in signaling in both the gut and the brain. And so we also, and so with skin cancer and breast cancer, specifically, there's been association with certain metabolites that are thought to impact local inflammatory processes, both in the skin as well as breast tissue that could be exacerbating those conditions.
What about other neurologic diseases like Alzheimer's disease or Parkinson's disease, both of which are increasing in frequency?
For sure. And so both of the metabolites that may be impacting the tissues or the specific cells themselves, as well as microbial signals that are impacting the inflammatory processes associated with these diseases are topics for research right now, including with our group. We have a few projects involving Parkinson's, Alzheimer's, and autism.
Thanks.
I have a question, Dr. Oswan, if you let me ask one little question. So, Dr. Petrosino, so what about the food that in the daily basis we eat? So is there any correlation between the type of microbiome that we might have, and is there any potential predisposition for some type of tumors?
Well, so we heard a little bit about today, and so I think the idea, the handshake with specific bacteria, so specific taxa have are less well described. But if you eat a healthy diet, so you're looking Mediterranean diet with healthy oils as well as fruits and vegetables, that tends to produce a more diverse gut microbiome itself, and in most cases, that means you have fewer inflammatory bacteria that are present, which is normally what you want for a healthy state. And as we heard in the last talk, sugars, acidic foods, not in this terms of citrus, but acidic foods or foods that actually neutralize the acidity of your gut can both have negative influences on the microbiome that could lead to loss of diversity.
So I have a question as well. I've probably seen a couple hundred, I know I've seen between 200 and 300 people over the last 8 to 10 years under the age of 50 with colorectal cancer. The vast majority do not have family history. We know they're not genetic because we test all of them. Fat, thin, short, tall, Hispanic, Black, white, Asian, across all lives. And we published a paper looking at Hispanics in Texas. Yet we really don't have a good epidemiological study now trying to link a certain kind of diet, hormones, antibiotics, and the meat we eat, or are using too many antibiotics, for example. We've touched on a few things, but I think there are also a couple things have not been studied, at least to my satisfaction. For example, everybody raise their hand if you ate food off the floor when you were a kid. We all did. Right? Is it possible that with the clean freak stuff that's going on now, the Purell everywhere, who had Purell in their house when they were a kid? Nobody did. Is it possible that it may not just be a function of what we're eating, but also how we're living our lives in general? Are there any data to show that you're excessively sterile compared to what we were 20, 30 years ago?
Yeah, that's a wonderfully framed question too. So there's been a lot of thought and research towards what the hygiene hypothesis is in terms of how Western practices for just eating and raising our children and how we conduct our own hygiene is having a negative impact on the microbiome. I can give a whole talk based on that, and not even research that we've done, but just what's out there. But one quick vignette is that there have been studies of pristine cultures, Amerindian cultures in South America, and also tribes in Africa who haven't been using antibiotics and the products that we just talked about, and look at the microbiomes of those individuals and comparing it to those, Western civilizations, and they're drastically different. They're much more diverse in terms of the taxa that are present. There are bacteria that are there that we don't have anymore. And so they're even taught, Marty Blazer, there's a book you can buy on Amazon called Lost Microbes that tells it's a very tractable story for the lay reader in terms of just exactly what you're talking about, how our practices in Western civilization has caused us to lose certain microbes. And even aggressively, like we are afraid of Helicobacter pylori for the most part. For example, it's the organism that is associated with peptic ulcers. This organism is present in these tribes, and they are perfectly healthy, don't have gastric ulcers at all. And it's thought that potentially, like some of the practices, even the foods that buffer the acidity in the stomach, has allowed the organism to travel into the stomach, and it's ectopic there. It doesn't belong there. And so the thought is that if you eat a lot of fiber, these cultures, one thing that they do with their diet, they eat 10 times the amount of fiber that we do, if not more. And it's thought that these practices keep the gastrointestinal health in balance and keeps the organisms diverse, keeps them growing where they're supposed to grow, and leads to better health.
We have a lot of questions from the audience right now. So we have a question that is mostly for Dr. Yoon by Tanya Matar. So what about diet drinks? Do they have the same impact like sugary drinks? We cannot hear you, Dr. Yoon.
Yes, can you hear? Right. I think
Yes.
that's a really important and interesting question, because now people switch to diet, and more people drink diet because people think that sugar drinks is bad. And honestly, I didn't research on that one yet, but maybe next project will be. But in terms of when I look at the human data, there is not much strong the data in terms of diet and cancer at this moment. There is some study in rat or mouse, but one thing I can say is that there is a study that actually artificial sweetener can actually change the microbiome, which is not surprising, and that actually also increase the obesity and pre-diabetic phenotype. So there is a definitely change of a microbiome, but in terms of cancer, we are not that sure about the causal link.
Okay, good that you talked about the artificial sweeteners. Actually, someone also asked, same person also asked if you know of the safest commercial sugar to use, whether raw brown sugar, stevia, or monk fruit.
Oh, you mean that it's okay to fruits in fruits,
Mm-hmm.
right? So I
Right.
think yes, I think also that is another popular question, because I really encourage to eat fruits. But the difference is that fruit has a lot of other things, fibers and other minerals. So when you eat fruits, you cannot absorb very quickly, like a complete pure sugar in liquid. So first of all, you cannot eat, one equivalent to Coca-Cola 12 ounce is like 7 apples. You cannot eat the 7 apples at once, right? So I think I would encourage the having fruits, because there's not only the sugars, but there are other really good things. So I think you shouldn't confuse with the sugary drinks with the fruits, sugars in the fruit.
Thank you, Dr. Yoon. We also have a question by Gloria Tenenbaum. Is there any benefit to adding probiotics to our daily diets?
I think maybe Joe can answer that one.
Yeah, so I get asked, and I apologize in the background. I have dogs there; it's kind of going nuts. I get asked all the time, do I take a probiotic? And the answer is generally no. And the reason is because it's just my own personal habits. But and then the other reason is that up until recently, the probiotics that are available over the counter most of them have not been what I would say would be up to snuff. They don't have the number. They didn't have the number of live organisms, or even the organisms that they said that they were in the box actually in the box or in the capsules. But I would say also, as we've had a renaissance in terms of understanding the microbiome with health and disease, there's been a greater effort by probiotic producers to create probiotics and probiotic foods that are much more robust. And so if you're interested, if you have, I would say if you have upset stomachs, if there's some sort of GI issue going on, that they can help for sure. But if you're normally, if you're okay and you're eating a good diet, there really isn't a need to supplement with probiotics at this time.
Okay. Thank you so much, Dr. Petrosino. We also have another question by Ethan Lostspring. What about the effect of sugar alcohols on cancer microbiome?
I mean, sugar alcohol, like a sorbitol, or that is not sugar alcohol. It's like alcohol itself. You mean alcohol, right? Mm-hmm. I think alcohol is pretty well known to the link to the many of cancers, including colon cancer. And maybe the pathway could be different, because maybe Ben can explain better than me in terms of alcohol and cancer. Ben?
There is, I can speak about a pancreatic cancer. Clearly, if you drink a lot, you get pancreatic inflammation. That's the risk factor for pancreatic cancers. So that's one thing. As far as colon cancer, there is, I believe, the relative risk, it's like 1.2 or something like that for alcohol and colon cancer. So it's not, it probably is a risk factor, independent risk factor, but not a particularly high one. Is it the alcohol itself or the sugary stuff in the alcohol? Is it a direct effect or metabolic effect? I don't think anybody knows, but it is a risk factor, but it's not one that we think of as being as strong as liver cancer, for example, when you get cirrhosis from drinking.
Thank you so much, Dr. Musher. I have a question that maybe for the general audience here, for all panelists. Are there any guidelines of how much sugar you should keep under?
I think nobody knows it. I mean, to me, I wouldn't say that you shouldn't restrict completely, like any dessert or cake occasionally. But I think as long as you keep avoiding the sugar drinks, I think that's the key. And other sugary stuff, I don't think you have to completely eliminate. I mean, if you can, it's good, but I don't think also you don't want to remove the joy of eating some good sweet stuff. So I think the guideline is that, for men, don't more than 9 teaspoons, and woman is no more than 6 teaspoons. But who knows, right? So I think do your best to try to keep it down and not be addictive. And that's my personal opinion. But other people can comment on that.
Dr. Yoon, would it be better to eat a cookie or drink a can of Coke?
I think that's
or ice cream.
Ice
cream.
Ice cream is ice cream. Maybe keep into maybe 1 scoop, not every day, maybe, right? Because liquid, as I mentioned, that could be more powerful to increase the sugar concentration in our gut. I think that's the key.
Here we have another question, a different topic. So if you have a younger sibling that has had colon cancer less than 30, should you get a genetic test? Are there any genes that predispose you to colon cancer?
Kathy?
So the answer to that.
Yep, I'm happy
Kathy, yes, absolutely. Thank you.
I'm happy to take that one. So the answer to that question is absolutely yes. If someone's diagnosed with colon cancer under the age of 30, that's definitely concerning that there could be an underlying genetic mutation running through the family. Now, ideally, we would want to test the person who has cancer first so that we could try to determine if the cancer is indeed linked to one of these underlying syndromes. And then if we did identify a mutation, we could test other family members to identify who's at an increased risk so that we could be more proactive and implement screening at younger ages. The good rule of thumb for colon cancer, if somebody's diagnosed at 50 or under, then they or their family members should consider genetic testing. That's unusually young enough to warrant genetic testing. or if there's multiple people in the family who have had colon cancer. Some of these syndromes also increase the risk for endometrial or uterine cancer. So if a family has those patterns of cancer, then you would also want to consider it. But absolutely, if somebody's diagnosed at 30.
Okay. And to follow up with that question, can someone who is healthy, as in they eat well and they exercise, still contract cancer or other diseases due to genetics?
Sure. I think we don't understand all of the different factors that play a role. So I always talk about, genetics is just one piece of the puzzle, and we don't really understand all of the other pieces yet. But I absolutely have patients who, think they've done everything right and they've still developed cancer. Even patients who don't have an underlying genetic mutation who think they're doing everything right can still develop cancer. So absolutely. And the flip side to that is just because you have a mutation doesn't mean you're going to get cancer either. Many people who are born with one of these underlying mutations never actually develop cancer, and we don't quite understand that either.
Yeah, to emphasize.
If I can add one quick point, I think a lot of this is common sense, at least at this point. We can't escape our genetics, not yet at least. We can modify what we do on a daily basis. We'll ask them what they can do. Don't smoke cigarettes. Don't drink too much. Stay relatively active. It doesn't mean you have to run marathons. Just stay on your feet. Take walks. Avoid sugary beverages. I always tell my patients, you want to lose weight, you want to make yourself healthier, cut out sugary drinks altogether. That's not a difficult one. The bowl of ice cream has protein and fat in it, which are actually good for our bodies. I'm not saying eat ice cream all day long, but if you have to choose. So for the time being, I don't think there's a lot of science to tell us what we should be eating or drinking. More fruits and vegetables, fewer sugary drinks, don't smoke, don't drink, stay active. And then at a certain point, we can't escape things that happen to our bodies over time, not yet at least. The science is important. It might lead us to some more modifiable behaviors that we can either avoid or partake in. But for the time being, I think that's where we have to live our lives in a common sense manner based on what we know. And also, screening, right? We do have science for screening for certain cancers at certain ages, and we should follow those as well.
Dr. Musher, can I ask you a question about your thoughts on taking aspirin to try to lower the risk of colorectal cancer? Is that something you recommend people do, or what is your current recommendation with that?
Right. So there have been studies looking at this, and it does appear that over large populations, aspirin does decrease colon cancer risk. It appears specifically to decrease a risk of PIK3CA mutated colon cancers. There are also other benefits of aspirin, and there are downsides too. I've debated this myself. I'm now 47 years old. I'm going to start taking a baby aspirin once a day. I'm not saying other people should do it as well, but I think the data are there enough for certain diseases, including cardiovascular disease and colon cancer, that I think it's reasonable to consider, but it is not a universal hard recommendation at this point.
What you say you're 47, so what's the age that you would recommend someone? I mean, would you recommend a 30-year-old start doing that, or would you say 45 to 50 is, I mean, is there data to guide that yet?
Not that I know of. Someone else can tell me if they know of any.
I don't know either.
Listen, I think although young onset colorectal cancer is increasing pretty dramatically in incidence has been and will continue to over the last 15, 20 years, it is still uncommon. And I think we have to know that. I mean, Chadwick Boseman, deaths of young people who have these, bring these things to light, but thankfully this is still an uncommon condition. It's still much more common in older people, although incidence decreasing because of screening that picks up polyps before they can even turn into cancer. So I think there are other benefits taking baby aspirin when you're older anyway. I don't think it's anything we can recommend for younger people at this point.
I have another question here.
A very quick, I mean, if you don't mind, I'm sorry for interrupting, but I wanted to ask Dr. Petrosino or even Musher, do you think we are in a prime, or how far are we from investigating potentially those germs that reside in the colon that represent the microbiome, and how informative, and I would say, how can that inform a patient potentially that the patient might have a predisposition for colon cancer, pancreatic cancer? Do you think the expression of those germs might in the future inform a particular risk?
I can, from a potential standpoint, I could say that there are a number of studies that have shown that if you add microbial signatures to current diagnostic or blood markers for cancer and other type of colon cancer as well as other types of cancer, it can increase the sensitivity of those tests or increase the accuracy of those tests. As far as being a sole indicator, that's probably a little bit further. We're a little further away from something like that. This is my experience.
Gustavo, I have a question for you.
Yes.
We haven't said anything about hematologic malignancies, yet they're among the most common in younger people.
Yeah.
What can we do about those? There's no way to screen for them would just be by a routine annual examination. But
So it's a very, very interesting question, Dr. Osborne. I think one of the dramatic changes that we have in malignant hematology is potentially the same kind of recapitulation that we're doing for medical oncology, in the sense that, you know, even being older, we can find significant amount of people carrying germline predisposition syndromes. Actually, that paradigm that those diseases are observed only in patients who are younger is not anymore certain. The other point is syndromic. So you don't need to have typical symptoms, typical signs, even clustering of families. You can have those mutations in low penetrance, inducing disease, and they have implication for selection of donors if the patient is going to be subjected to bone marrow transplantation, and even for therapeutic implications. One of the examples that are extremely important that we see in older patients with germline predisposition, like DDX41, that we normally treat with Revlimid. So it's impressive. The responses are almost 90%. So I think, you know, things have changed, and we should continue to look for these germline predispositions in other individuals.
Okay. I have a question here. If you do endurance sports like triathlons, should you not have energy gels with high levels of sugars, drinks like Gatorade or Coca-Cola?
What was the question? Is that whether the energy, like get those, what's that? What the energy sports drink is safe, or that's the question?
Right. Mm-hmm.
Yeah, I don't think it's safe. It's basically sugary drinks, and they have exactly high concentration of sugar when you look at check the sugar thing. So that's also be careful. We have to be careful about those sports drink as well. So the best way is to drink water. I think that's the thing. And I really hope that Baylor take an initiative to get rid of all the sugary thing in the cafeteria, because they get for free when you have the thing. And I just feel that most of people, actually 80% of people seems like go to the free Coke or free soda instead of taking water bottle. So yeah, it's very big. So
Dr. Yoon, but wouldn't you agree that just drinking some sugary drinks during a race, and not chronically, but just during a race to keep your carb level up, that would be okay, right?
Yeah. To get the energy, to give the energy boost, right? They maybe.
I think the point is this is probably more about repeated continuous exposure than having
Yeah. And also, I think if we don't drink crazily with one time, maybe, if we use kind of one and then have an interval, I think that maybe also reduce the saturating all the transporter in the small intestine. Yeah.
Thank you so much, Dr. Yoon. There's also another question that I think this comes from my family. An older sibling with osteosarcoma, should younger siblings seek genetic testing?
I can take that one again. And, probably not if it's an older person in the family and an isolated case. There are certain osteosarcomas that can be related to genetic syndromes, but typically we would see other characteristics in the family that would concern us that there was something genetic going on. So I think that would warrant kind of a thorough family history, learning a little bit more about the family history before we could determine if that situation warranted genetic testing or not.
Thank you so much, Kathy.
If I could add one point about genetic testing. So we have really good genetic counselors at Baylor, and I'm not just saying this because Kathy's on the line. The way that I see the genetics thing is the following. If they're younger people with cancers, what I typically do is I just offer them genetic counseling. We know there's data on this. Physicians are notoriously bad at eliciting family histories from patients, and patients are notoriously bad at knowing their family histories unless they dig into it. One nice thing about getting genetic counseling is that patients are forced to find out what's going on in their families and to talk to someone who knows what the data are about screening. So I offer people genetic counseling when they want it, or I think it's indicated, and genetic counselors do the heavy hitting when it comes to gathering that information. So to answer your question, if there's ever any doubt, ask to see a genetic counselor if your doctor oncologist thinks it's reasonable, and if you don't have one where you are, then ask for one in an academic center. I think it's a very important thing to do.
Thank you so much, Dr. Musher.
Osteosarcoma being such a, I would say, especially the location. So for my own education, so what is osteosarcoma? And what are the mutations that normally are linked to a germline predisposition? Anybody? Kathy, I think you have to unmute.
Sorry, when I think of any type of sarcoma, I typically think of P53, Li-Fraumeni syndrome, and we would typically see lots of childhood cancers in those families, which is why I would typically say an isolated osteosarcoma at an older age is not likely going to be linked to an underlying germline mutation.
I see.
Yeah, thank you so much, Kathy. I have a question by Roger Maldonado. Samples of best food to promote healthy gut and good bacteria.
I missed the first part of that. What promoting what? What was the question?
Yes, samples of best food to promote healthy gut and good bacteria.
The leafy greens are traditionally where people go. I mean, all fruits and vegetables are good, but the leafy greens and the greener the vegetable, usually the better off you are. Fruits are okay too, but the most fiber and the most nutrients that bacteria like come from the sort of your kales and your spinach and all the ones that people hate to eat, but unfortunately those are the ones that bacteria love.
Thank you so much, Dr. Petrosino. I have a question by Gloria Tenenbaum again. What can we do to get insurance companies to cover screening for breast, colon, and testicular cancers in people younger than 40?
It's a good question.
I think
Dr. Osborne.
Say the question again.
Yeah, sorry. What can we do to get insurance companies to cover screening for breast, colon, and testicular cancers in people younger than 40?
Yeah, I think we can argue with the insurance companies, and some of them will cover it if you have a reasonable explanation for why. And sometimes you have to argue with them, but I think that just comes down to the physician talking with the insurance company to convince them it's needed. Obviously, if you've been proven to have a genetic mutation, then that's relatively easy.
I can speak a little to that as well, especially from a breast cancer perspective. For unaffected women, there are risk models that can help us to calculate their personal risk to develop breast cancer. And oftentimes we can submit that to insurance if they have a high risk to help push through approval for screening. And we can explain, if the age of breast cancer in the family was 40, then we want to use that 10-year rule of thumb to start the screening at age 30. And so we can use some of that information in our notes to try to push it through the insurance companies. I have found that if you have a specialist who writes that note, sometimes it can go a long way with the insurance companies.
Okay, well, thank you all of our speakers and panelists, and thank you to our audience for all the questions and for your time. This concludes today's event on young adults and cancer. And yeah, y'all have a good night.
Thanks, Victor.
Thank you.
Thank
Yeah.
you.